The title of this study is Relationships of Behavioral and Physiological Symptoms of Pre-Slaughter Stress to Beef Longissimus Muscle Tenderness. The group members are Creston Ridlinger, Graham Rossi, Anna Wachter, and Alexis Zaborniak. Cattle can be negatively affected in the handling and transporting of the animal prior to slaughter. Hands-on moving of cattle can stress the cow out and cause behavioral changes in the animal. Thus, this can affect the meat quality. Animals react to the routine pre-slaughter handling differently, so this can give a wide variety and range of different behaviors that can be tested. Animals sometimes do not show a change of behavior when they're being handled, but most show behaviors such as nervousness, fear, running from the handler, balking, avoiding the handler, vocalization, aggression, or any physiological change or sign of stress in the cow. When the cow is stressed, epinephrine and norepinephrine, which are neurotransmitters, are released. When epinephrine and norepinephrine are released, they cause increased blood glucose levels, an increase in the breakdown of glycogen in skeletal muscle cells, breakdown of fat in adipose tissue, and they cause dilation of blood vessels in skeletal muscles and cardiac muscles. All of this just basically means that there will be an increase in the heart rate, and there's a breakdown in glucose and fat in the skeletal muscle cells. All of the effects added together lead to the decrease in meat tenderness. It's also documented that pre-slaughter stress affects muscle color, firmness, pH, and water retention in the muscle. The study Relationships of Behavioral and Physiological Symptoms of Pre-Slaughter Stress to Beef Longissimus Muscle Tenderness was conducted by Gruber, Tatum, Engel, Chapman, Belk, and Smith to gain further insight into the relationships of pre-slaughter stress on the quality of meat. The study was conducted on 156 calves from a single herd from the Eastern Colorado Research Center. The herd was made up of 79 steer calves and 77 heifer calves. The calves started out by being immunized and weaned. They were then sent off to Colorado State University's Beef Research Feedlot for the finishing process. The cows were separated by gender once they arrived to the feedlot and went through a period of acclimation before any tests were run. The calves were weighed individually to determine weaning weight, and then they were implanted with trenbolone acetate and estradiol. These together would increase muscle growth and appetite. The calves were then separated more, now into 16 different pens for finishing, and all 16 pens were fed the same diet, and they were fed ad libitum, which means that they had free reign of the food. Finally, the cows were almost ready for slaughter. They were implanted once more with the same substances as before, which were trenbolone acetate and estradiol. There were behavior responses collected in 3 different situations. The first one was to have a human go in the pen with the animal and take a score as to what the behavior of the animal was when that human entered the pen. The second was a reaction of the animal being confined in a squeeze chute. And the third was a reaction of being transported to the packing facilities. These animals' reactions were recorded from a scale from 1 to 5. 1 being calm, 2 restless, 3 is nervous, 4 flighty, and 5 aggressive. Blood samples were then collected before slaughter, after slaughter, and post-mortem. Also, a Warner-Brotzel shear force score was acquired. These scores together, along with other testing techniques, allowed scientists to determine the effect of handling on post-slaughter meat quality and tenderness. Now we will discuss more methods and materials in this study.
So within 1 month of the slaughter, the cattle were evaluated on a scale that Alexis talked about, 1 to 5, by 2 separate evaluators who did not discuss with each other so that the scores were unbiased. And then those 2 scores were averaged for one final score. The scores of each of the cattle in each pen were also calculated, and then one final number was found for each pen. The animals were scored within 3 hours so that there were no outside external stimuli for effect on the score. The cattle were all scheduled on 4 separate days within 90 to 111 days of the implant of estradiol that, again, Alexis discussed before. So before each designated slaughter day, 4 to 5 of the animals representing each sex and sire were tested in a hydraulic squeeze chute, and the behavior was recorded when the sides were pressed together in the chute. Blood was drawn quickly after this, and the collection in 10 milliliters non-heparinized tubes for cortisol collection. The heart rate and the temperature of each animal were also recorded for further data to see other psychological effects of the stress caused by these stimuli. The blood sample collected was about 10 milliliters, and it was allowed to coagulate at room temperature for about 4 to 6 hours and was then centrifuged. The animals themselves were then transported about 64 kilometers to a commercial facility slaughter site, and they were tested again for their reaction to human presence in the holding pens. So again, there were 2 evaluators for this portion. They went in separately and did not speak to each other, rated the animals, and then averaged the score for each animal and for each pen. The animals were then slaughtered, and blood was taken and chilled from the carcasses. The carcasses themselves then went through a series of electrical stimulation, which actually you use electrical stimulation to allow for more meat tenderness because it can rip the muscle apart, making the meat softer. So they went through a 16 volt, a 20 volt, a 24 volt, and a 28 volt electrical stimulation, and then they were sent to cool off, being sprayed for 2 minutes every 10 minutes with just some cool water. And then the marbling assessment were averaged for each carcass, with 300 being only slight marbling, 400 being small marbling, and 500 being modest marbling. Also, the tests were completed for the L, the A, and the B, which is something that's used to measure color. So that is where you take the spectrophotometer, calibrated with black and white tiles in order to compare, and then they were operated in a 2 degrees Celsius atmosphere. The strip loin was removed from each side of the carcass, or from the right side of the carcass of each carcass, and was transported to the CSU meat laboratory, where they were randomly assigned different sampling techniques to identify different longissimus muscle. After the aging process, the samples were cooked on an electric conveyor grill for a constant time of 6 minutes at 163 degrees Celsius for the top and bottom heating plates to achieve a peak internal temperature target of 71 degrees Celsius. Then the Werner-Bratzler shear force test was used on each of these pieces of meat. And this is an example of one of the machines. Basically, it's a sharp, thin piece of steel, and you put a piece of meat in the middle, and you put pressure on the thin steel, and that's measured in either kilograms or pounds, depending. And depending how much weight it takes to shear off a piece of the meat can show you how tender the meat is. So usually a lighter weight tends to mean more tender meat, and they use this to decide how palatable the meat's going to be in the end. So you usually want it to be a lighter weight. So now let's hear from Graham Rossi about analytical procedures and results.
Multiple analytical procedures were used at the end of this study. for both pre- and post-slaughter samples. Serum samples analyzed at the Department of Animal Science at New Mexico State University. These were analyzed for cortisol concentrations, creatine kinase levels, plasma glucose levels, and plasma lactate levels. Furthermore, we also wanted to analyze the pH of our meat samples. A healthy pH is around 5.5 in normal meat. To do this, we removed a piece of the longissimus muscle that was closest to the anterior end of the strip loin. After 3 days post-mortem, 3 grams of that meat was sliced, and then deionized water was added until it was properly homogenized, and then the pH test was conducted. A series of statistical methods were employed in order to compute the correct results from all 3 categories that we were looking at. The 3 categories were behavioral scores, hormone concentrations or physiological variables, and muscle quality or meat quality. For all of these, we used the Remmel-based mixed effects model. We used this model because in our subject group, we were dealing with cattle from multiple sires and sexes. The Remmel-based mixed effects model estimates components of variance in unbalanced data. When looking at the WBSF scores, we used an ANOVA model to compute these results. The ANOVA model compared the means of the meats, and it told us if they were significantly different for each of the stress levels. Later, we used the Kenward-Roger approximation, which was used to calculate the correlations between physiological variables and the transportation variables. This was conducted at a P of less than 0.05. On the day of the experiment, the following conditions were present. The weather was dry and mild. The temperature was between 7 and 26 degrees Celsius. The loading onto the trucks took between 4 to 6 minutes for each of the groups. And the travel time to from the feedlot to the slaughterhouse facility was between 64 to 90 minutes. First, we can discuss First, we'll look at the behavioral results and the scores. The behavioral score ranged from calm to flighty. When humans appeared in the pen, 39% of cattle were calm, while 61% were considered anxious. When cattle were subjected to physical handling, 30% were calm and 70% were anxious, so it increased with physical handling. Next, we were looking at the correlation between physiological stress indicators and behavioral stress indicators. We found a positive correlation between high stress scores and high heart rate, respiration rate, rectal temperature, serum cortisol level, and serum epinephrine levels. All of the correlation statistic, or R values, were above 0.29 for each of the physiological indicators. However, the post-transportation behavior score was not correlated with circulating glucose and cortisol levels. The R values, which is the correlation statistic, were below 0.01 for both the glucose and cortisol. On the other hand, the post-transportation score was positively correlated with lactate levels and creatine kinase levels. The R statistic was above 0.28 for both the lactate level and the creatine kinase. If we look at our results, this is just a general overview of all of our data. Up here we have the data, which follows the cortisol levels and the epinephrine levels. On the far right side, it shows the post-transportation behavior. You can see that at calm, restless, and nervous, the cortisol and epinephrine levels have very little variation. Therefore, they're not significantly different. We can also look at the longissimus muscle pH scores. In general, we want the pH score of beef to be between 5 and 5.5. In our data sheet, it shows that between calm and nervous behavioral scores, the pH did not vary greatly. They all remained between 3.8, 5.38, or 5.37. Cattle with significantly elevated stress responses produced tougher longissimus muscle cuts than calmer cattle. In general, all the cattle categorized in the nervous behavioral score had longissimus muscle cuts of 3.72 kilograms, whereas cattle categorized in the calm behavior score had longissimus muscle cuts of 3.49 kilograms, and that was significantly lower. Additionally, carcass pH indicated that there were no dark, firm, dry cuts there. Therefore, our meat was generally healthy without considering the effect of cortisol and/or other stress hormones in the meat. And now I'm going to pass it over to Creston so he can discuss the discussion.
Okay, so in this study, we showed that the correlation between stress of handling, transportation, slaughter, on the meat quality of the cattle measured with the longissimus muscle WVSF, which was described earlier, and the stress from the handling, transportation, and slaughter activates two pathways. One is the hypothalamic pituitary adrenal axis, the HPA, and the sympathoadrenal axis, the SA. These are both activated when the cows are put under stress. The HPA stimulates secretion of the hormone ACTH, or the adrenocorticotropic hormone. This is secreted from the anterior pituitary gland and causes the production of cortisol by the adrenal cortex. When cortisol is released into the blood, it stimulates plasma glucose levels that activate protein catabolism, or breakdown, in the skeletal muscles. In the study, they did not find this to be any correlation to the decrease in meat quality. The second pathway is the SA axis. When this is activated by acute stress, it releases two neurotransmitters, epinephrine and norepinephrine. They are released from the adrenal medulla oblongata. Epinephrine activates beta-adrenic receptors, and this causes an increase in plasma, hepatic, and muscle glucose. It also increases the lactate concentrations, heart rate, body temperature, and respiration. Depletion of the muscle glucose is also a known factor for decreased muscle quality, and suggests that increased pre-slaughter stress causes the activation of the SA and also reduces meat quality. In conclusion, this study shown that plasma epinephrine concentrations, which is caused by stress, can cause heart rate increase, rectal temperature increase, and plasma lactate concentration increase. And the plasma lactate increase is the cause associated with higher muscle pH and a higher longissimus muscle WVSF and lower meat quality. The study also shown that nervous cattle from non-physical human contact caused an increase in stress, and this caused lower quality meat compared to cows that were more relaxed with the same situation of the non-physical human contact. And the same was shown in the study with the handling and transportation of them. And the reference to the article is there.